Welcome to Science Hub. Today we're diving into a groundbreaking study that sheds new light on how gut bacteria, specifically E. coli, spread from one person to another, a feat that was previously only possible for viruses. For the first time, researchers at the Wellcome Sanger Institute, along with universities in Oslo, Helsinki, and Finland's Aalto University, have estimated the transmission efficiency of gut bacteria. This study, published in Nature Communications, could transform our approach to combating bacterial infections. The research focused on 3 major strains of E. coli prevalent in the UK and Norway. Notably, 2 of these strains are resistant to multiple antibiotic classes and are leading causes of urinary tract and bloodstream infections in both countries. By improving surveillance of these strains, we can better direct public health responses and potentially prevent outbreaks of hard-to-treat infections. Understanding the genetic factors that facilitate the spread of E. coli could lead to more targeted treatments and reduce our dependence on broad-spectrum antibiotics. The methods developed in this study could be adapted to investigate other bacterial pathogens and enhance strategies for managing invasive infections. A key concept in this research is the basic reproduction number, known as R0, which estimates how many new cases one infected individual can generate. Traditionally applied to viruses, R0 helps predict whether an outbreak will expand or decline. Until now, researchers couldn't assign an R0 value to bacteria that typically colonize the gut, as they often reside harmlessly within us. To overcome this challenge, the team combined data from the UK's baby biome study with genomic information from E. coli bloodstream infection surveillance programs in the UK and Norway. Assigning an R0 value to bacteria opens the door to a clearer understanding of bacterial infection spread. It also helps identify which strains pose the greatest threat, guiding public health strategies to better protect individuals with weakened immune systems. Co-first author Vani Oyala from Aalto University noted, "With a large amount of systematically collected data, we were able to build a simulation model to predict the R0 for E. coli. To our knowledge, this was the first time for any bacteria living in our gut microbiome." Professor Jukka Korander, lead author from the Wellcome Sanger Institute and the University of Oslo, added, "Having an R0 for E. coli allows us to see bacterial spread across populations more clearly and compare this to other infections. Now that we can see how quickly some of these bacterial strains spread, it is crucial to understand their genetic drivers." This research marks a significant step forward in understanding bacterial transmission, especially antibiotic-resistant strains. By determining the R0 for E. coli strains, we can better assess their threat level and guide public health efforts to protect vulnerable populations. This approach isn't confined to E. coli alone, but can extend to other pathogens, offering a powerful tool in battling bacterial infection spread. Don't forget to subscribe to Science Hub for more fascinating explorations into the world of science.